All right, ladies and gentlemen, here we are. We're going to talk about transcription, right? Step one of a two-step process to go from mRNA to protein. This two-step process is what's going to take us the final length of the way from genotype to phenotype. So, transcription is the process of forming a strand of RNA from a strand of DNA. Okay? So this is the actual process of making that mRNA that's going to travel out of the nucleus and to the ribosome. And of course, because DNA is in the nucleus, this process of transcription occurs in the nucleus. Now, the cell must make RNA to send to the cytoplasm, right? Because remember, DNA doesn't leave the nucleus. So this RNA copy acts as the messenger and leaves the nucleus, something DNA cannot do. It is a faithful copy of the DNA. It should be an exact copy of a certain gene on DNA. Now, ultimately, the enzyme called RNA polymerase, not to be confused with DNA polymerase, the enzyme involved in copying DNA, catalyzes this reaction. So DNA polymerase makes new DNA, RNA polymerase makes messenger RNA. And ultimately, the purpose of transcription is to copy just a single gene at a time because remember, a gene codes for a protein. And of course, DNA is one long molecule, so we have to beg the question, where does one gene end and the next gene begin? This is where we get into this idea of a promoter, but we're not going to talk anything about promoters. That's a upper level science topic, so we're going to cut off right there, as well as terminator. Just know that there are regions of genes that distinguish one gene from the one next to it. All right, let's look at the steps of transcription. And notice again, I've had this diagram on this slide and the last one now. Double-stranded DNA. We have a bubble, just like we did in DNA replication. But instead of making two new strands of DNA, we're just making this one single-stranded piece of mRNA, right, from a single gene. So, RNA polymerase binds to the site on DNA, on a DNA molecule called this promoter. This is where the gene starts. So RNA polymerase binds where a gene starts. RNA polymerase separates the two strands of DNA, meaning it has its own helicase activity, right? Remember, helicase was that individual enzyme that would unwind DNA for copying. Well, RNA polymerase can do that all on its own. Only one side of the DNA molecule is used as a template, which we can see happening right here. New nucleotides are inserted according to the base pairing rules. We're still following base pairing rules as well. So when transcribing RNA, adenine pairs with incorrect. We are not going to pair it with thymine because remember, in RNA, thymine does not exist and it's replaced with uracil. And cytosine in RNA pairs with, you're right, guanine. That's the same as DNA. And this is going to continue until that terminator, that endpoint, is reached of the gene. And as RNA polymerase moves along the molecule, hydrogen bonds between the DNA strands are reformed. So this bubble, as RNA polymerase moves in this direction, this bubble is going to close behind it, and the hydrogen bonds between nucleotides will reform. And ultimately, you get this one single strand of RNA that is made. Now, remember the purpose of transcription. The purpose is not to copy the entire length of the DNA molecule, but to copy small portions, a gene's worth, if you will. And that's what's going to be sent to the ribosome, and that's what codes for a protein. All right, we're going to take a little segue here, folks. We are going to skip from this slide number 45, all the way to slide number 49, the genetic code. And on your study guides, you should have been right here at transcription, okay, writing all this stuff down. And then we're going to get to this part, right? And we're going to skip this part. You'll come over here. And remember, if yours looks like this, just click these lines. We're going to come over here, and we're going to find the genetic code. And we'll click that. And that's where we're going to pick up at. So, Proteins are made by joining together long what? Correct. Chains of amino acids. The monomers of proteins are amino acids. And it's in fact the order in which these amino acids are joined that determine the type of protein that's made. Just like the order of nucleotides in DNA determine the genes in that strand of DNA. The language of mRNA instructions is called the genetic code. The genetic code, we know right now, is read in a series of 3 nucleotides at a time, and we call that series of 3 a codon. And again, a codon is just a series of 3 nitrogen bases that specifies an amino acid. So, codon codes for an amino acid. Genes code for proteins. Codons make up genes, amino acids make up proteins. All right, so let's do an example. Here's a strand of mRNA. This sequence would be read left to right, 3 bases at a time. Now, on Canvas, you have a codon chart. All right, you have, let's exit out of here. You have a codon chart, both a new one. Okay, this new one, these are all the amino acids around the outside, all 20 of them. And these are your codon, or I'm sorry, these are your nucleotides in the middle. And since you read nucleotides in 3 at a time, they're set up 3 at a time. You start in the middle, so if your first nucleotide's U, you start here. If the next one is an A, you go out to here. And then, if your last one's a U or a C, it's tyrosine. If your last nucleotide is an A or a G, it's a stop codon. Okay, so you read this one from the inside out. And then I also have for you the old codon chart. The old codon chart is not as user-friendly looking, but could also show up on the keystone. So, your first letter is on the left, second letter across the top, and third letter down the right. So it's going to go, let's say your first letter is a C. Okay, first letter is a C, and then your second letter is an A. So C and A bring us to here. Okay? And then if the third letter is a U, the amino acid is his or histidine. If it's a C, it's still his or histidine, but if it's an A or G, it's going to be glycine, or glutamine, GLN. All right, this one's a little harder to read and we'll take some more practice. Let's jump back into the notes. So keep those codon charts up and try to decode this series of 3, or I'm sorry, this gene that I've given you, this mRNA transcript. So the first thing you want to do, the easiest thing to do, is to break it up into its codons first. Break it up into its codons first. So now we have 3 codons. That means that this segment of mRNA should code for a set of 3 amino acids, and they are serine, histidine, and glycine. See if you can find these and how they code for those on your codon chart if you haven't already. So, hang on one second. I think we're going to skip over this as well. Yes, we are going to skip over that. And in fact, we'll end right there, folks. See you guys in the next one.